Take a look at your blood type card. A, B, AB, or O. That letter isn't decided by a special blood gene alone. It's written by sugar chains called glycans, stitched onto the surface of every red blood cell you have. Welcome to the glycan code, a layer of biology so information-dense that some scientists now call it the true frontier beyond the genome. Sugars aren't just fuel for your cells, they're a communication system. They coat your cell membranes. They dangle off nearly every antibody in your bloodstream. They decorate your hormones, and they thicken the mucus lining your gut and lungs. Almost nothing your body makes leaves the factory without a sugar label attached. So what exactly is a glycan? In plain terms, it's a chain of sugar molecules, sometimes just a few units long, sometimes branching like a tree, attached to a protein or a fat. Scientists call the complete set of these sugar chains in your body the glycome, and it's turning out to be one of the richest sources of medical information we've ever found. For 20 years, genomics dominated the diagnostic conversation. Sequence the DNA, find the mutation, predict the disease. But your genome is nearly identical in every cell and barely changes over your lifetime. Your glycome is different. It shifts hour by hour, reflecting exactly what's happening in your body right now, which is exactly what a doctor trying to catch disease early actually wants to know. Here's a single building block of that sugar code. A simple 6-carbon ring, the kind of molecule you'd recognize as glucose from a basic biology class. Individually, it looks unremarkable, but start linking dozens of these rings together, in different orders and at different angles, and you get something extraordinarily information-rich. Think of it as 3 overlapping codes of life. Your genome is the recipe book. Your proteome is the kitchen staff built from that recipe. And your glycome is the finishing garnish, except this garnish isn't decorative. It controls how proteins fold, how long they survive in your blood, and which cells they're allowed to talk to. Here's the number that gets glycobiologists excited. With just around a dozen common sugar building blocks, but multiple ways to link each one and branch it in different directions, your body can theoretically build over 1 million distinct glycan structures. DNA, by comparison, only ever links its 4 letters in a single straight line. Zoom into any single cell in your body, and you'd find it wrapped in what scientists literally call a sugar forest, the glycocalyx. This fuzzy coating is often the very first thing another cell, a virus, or your own immune system touches. Every interaction your cells have with the outside world starts with sugar. So before we go further, glycans are not decoration. They're a dynamic, real-time information layer sitting on top of your genome and proteome. And as you're about to see, reading that layer is opening up an entirely new frontier in how disease gets diagnosed. Here's a sentence that surprises most people the first time they hear it. Your sugar code may carry more raw information than your genetic code ever could. Not because sugars are smarter than DNA, but because of how they're built. DNA and proteins are built like sentences, one letter after another, in a single straight line, following a fixed template. Glycans break that rule entirely. A single sugar can link to a neighbor at 4 or 5 different attachment points, and it can branch off in multiple directions at once, like a tree instead of a sentence. Picture a single glycan as a small branching tree of sugar units, each branch ending in a different sugar with a different job. Add or remove just one branch, or flip the angle of a single bond, and you've created a molecule your immune system reads completely differently. To put the scale in perspective, your genome uses a 4-letter alphabet, read in one direction. Your glycome uses roughly 10 to 12 common sugar building blocks, each of which can attach in multiple positions and branch freely. That single difference explodes the number of possible words you can spell. You've already met one everyday example of this code, your ABO blood type. The A, B, and O antigens are literally different sugar caps built by different enzymes onto the same base structure on your red blood cells. One extra sugar unit is the entire difference between type A and type O blood. This is a good place to clear up a common misconception. Myth: my DNA test tells my doctor everything relevant about my current health. Fact: DNA tells you what you were built with. It says very little about what's happening in your body today, which is precisely the gap glycans are stepping in to fill. Researchers estimate that a single human gene, through alternative sugar decoration alone, can end up as dozens of subtly different finished molecules in your blood. Multiply that across the roughly 20,000 genes in your body, and you start to see why the glycome dwarfs the genome in sheer variety. Leading glycobiologist Ajit Varki has put it plainly: despite 3 billion years of evolution, life has never produced a single free living cell without a sugar coating. If evolution refused to abandon this code for 3 billion years, it's probably worth our full attention now. The takeaway for this chapter: because glycans branch instead of running in a straight line, they can encode far more combinations than a linear code ever could. That combinatorial richness is exactly why doctors are starting to look here for answers genomics alone can't give them. So where do these sugar chains actually come from? Unlike DNA replication or protein synthesis, there's no master template for building a glycan. Instead, every glycan is assembled step by step, inside a cellular factory, by an assembly line of enzymes reading the situation in real time. That real-time, enzyme-driven process is exactly why glycans make such powerful diagnostic clues. They're a live readout of your cell biology, not a fixed blueprint. If a video like this is helping the science click for you, a quick subscribe helps more of these come your way. The process begins in a compartment called the endoplasmic reticulum, where a starter sugar tree gets attached to a freshly made protein. That protein then travels through the Golgi apparatus. Think of it as a series of finishing stations, where enzymes called glycosyltransferases trim some sugars away and add others, station by station. Each of those finishing stations is run by a dedicated enzyme, and the amount of that enzyme your cell happens to be making at that exact moment decides the outcome. Change the enzyme, and you change the message. No gene mutation required, just a shift in what the cell's machinery is doing right now. Compare that to how your body makes a protein from DNA. A gene provides a fixed template. Ribosomes read it letter by letter, and the outcome is largely predictable and repeatable. Glycan assembly has no such template. It's more like improvisation guided by whatever enzymes happen to be on shift. Here's why that matters clinically. Consider a 52-year-old with early liver inflammation. Her DNA hasn't changed at all, but the glycosyltransferase enzymes in her liver cells have quietly shifted their activity, adding extra fucose sugars onto a protein called AFP. That single sugar shift becomes detectable in her blood long before a scan would show a mass. In that scenario, the fraction of fucose-tagged AFP in the blood, called AFPL3, climbing above 10% is associated with a roughly 7 to 10-fold jump in the risk of developing liver cancer within about 2 years. That's a sugar measurement doing serious diagnostic work. Zoom in far enough, under an electron microscope, and you can actually see this glycan forest, a dense, fuzzy coat bristling off the surface of every cell, roughly as thick as the cell membrane itself. It's not a thin dusting. It's a substantial, load-bearing layer of biology. So remember this: glycans are built live, station by station, with no fixed template, which means they capture what your cells are doing right now, not just what your DNA once encoded. That real-time quality is the whole reason they're so useful in a clinic. Every code can carry an error. When the glycan code goes wrong, the consequences range from a rare genetic disorder to some of the most aggressive cancers we know. Let's look at what happens when the sugar factory starts making the wrong products. Cancer cells are notorious for hijacking this system. As a tumor grows, it ramps up enzymes that pile extra sialic acid sugars onto its surface proteins, a process called hypergylylation. Those extra sugars aren't just clutter, they're a disguise. This isn't a side effect, it's an active survival strategy. The extra sialic acid sugars specifically engage inhibitory receptors called Siglecs on natural killer cells and macrophages, sending a "don't attack, I'm normal" tissue signal, essentially a molecular cloaking device built entirely out of sugar. Cancer glycans don't just hide from immune cells, some of them become brand new targets. Tumor cells can display an unusual sugar antigen called GD2, which is essentially absent on healthy adult tissue but abundant on neuroblastoma cells, giving antibody-based therapies a nearly cancer-exclusive sugar flag to lock onto. Drug developers have learned to engineer glycans on the therapy side too. Take obinutuzumab, an antibody used against certain leukemias. Scientists strip a single fucose sugar off its base, a technique called a fucosylation, and that one missing sugar dramatically boosts how tightly the antibody grips immune effector cells, multiplying its cancer-killing power. Cancer isn't the only place this code misfires. In autoimmune disease, Changes to the sugars on your antibodies can flip them from peacekeepers into troublemakers. In rheumatoid arthritis, for instance, antibodies often lose a sugar called galactose, which shifts them toward triggering inflammation instead of calming it. Quick pause. If this breakdown is helping you, take a second to hit like and subscribe. It genuinely takes a lot of effort to make videos like this, and your support keeps them coming. Doctors can already see this shift on a molecular map, where a healthy IgG antibody sugar tail typically ends in galactose and sialic acid, and inflamed, arthritic joints antibodies are missing those caps, a stripped-down sugar pattern that correlates directly with disease flares and severity. At the far end of the spectrum sit the congenital disorders of glycosylation, a rare group of genetic conditions where the sugar-building machinery itself is broken from birth. They range from mild to severe, and they remind us that when this system fails outright, the effects touch nearly every organ system at once. How do clinicians actually catch these? A decades-old test called transferrin isoelectric focusing separates a common blood protein by its sugar pattern. A healthy pattern shows a clean set of bands. A disrupted glycosylation pathway shows extra, abnormal bands, a sugar fingerprint of the underlying disease. The pattern across all three of these examples, cancer, autoimmune disease, and rare genetic disorders, is the same. When the sugar code breaks, the break shows up in the blood long before it shows up anywhere else, which is precisely why it's such fertile ground for diagnostics. So this isn't hypothetical. Future medicine, glycan-based tests are already sitting in hospital labs today, quietly informing real diagnoses. Let's walk through a few you might actually encounter. These aren't experimental research tools. Some of these glycan tests have been used in hospital labs for years. If you're finding this useful, tap that like button. It genuinely helps this kind of science content reach more people. Take CA19-9, a sugar antigen called sialyl-Lewis A that rises in the blood with pancreatic cancer. It's useful, but it's not perfect. Roughly 5 to 10% of people are genetically unable to make this antigen at all, so a normal result in those patients doesn't rule anything out. A real limitation clinicians have to keep in mind. Compare that to AFPL3, the fucosylated sugar tag version of a liver protein we met earlier. In patients already at risk for liver cancer, an AFPL3 level above 10% is tied to roughly a 7 to 10-fold jump in cancer risk over the following months, a striking amount of signal packed into a single sugar measurement. Here's how a clinician actually uses that number. A patient with chronic liver disease and an AFPL3 of 14% isn't necessarily told, "You have cancer." They're moved into a closer surveillance track, with more frequent imaging, because the sugar signal flagged elevated risk before a mass was ever visible. One of the more surprising applications is aging itself. Researchers found that the sugar pattern on your IgG antibodies, specifically how much galactose and sialic acid decorate them, tracks your biological age more closely than your birth certificate does, giving rise to what's sometimes called a glycan age test. In practice, two 45-year-olds can carry very different glycan signatures. One with a youthful, well-galactosylated antibody pattern, another, under chronic inflammatory stress, with a stripped-down pattern that resembles someone a decade or more older, the same birthday, a very different biological story. And of course, there's the test almost everyone has already taken, ABO and Rh blood typing. It works by mixing your blood with antibodies that recognize specific sugar caps. Clumping means the sugar is present. No clumping means it isn't. It's one of the oldest, most reliable glycan tests in medicine, running quietly in the background of every transfusion and surgery. It's worth being clear-eyed about where each of these tests sits on the evidence ladder. ABO typing rests on a century of rock-solid clinical use. AFPL3 sits on strong, FDA-recognized evidence for liver cancer risk. Glycan age, by contrast, is still largely a research and wellness tool, with promising but earlier stage evidence behind it. So the honest picture is this. Glycan diagnostics aren't science fiction, but they also aren't all equally proven. Some, like blood typing, are gold standard. Others, like biological age testing, are promising but still maturing, and knowing that difference matters. If today's glycan tests already sound impressive, the next wave of tools is aiming to read the entire sugar code at once, not just one marker at a time, turning glycomics from a handful of individual tests into a full-spectrum diagnostic map. The workhorse of this next wave is mass spectrometry glycomics, a technique that weighs and fragments thousands of individual sugar structures pulled from a single drop of blood, then matches each fragment's mass against a known library to reconstruct the complete glycan profile, all in one run. A complementary tool called a lectin microarray takes a different approach. It spots hundreds of different sugar-binding proteins onto a single chip, then floods it with a patient's blood sample. Wherever a specific glycan is present, it lights up against its matching lectin, a whole glycome fingerprint in a single grid. This next-generation glycan reading isn't only about disease detection. It also reshaped vaccine science. The spike protein on SARS-CoV-2 is so densely coated in glycans that researchers described it as wearing a glycan shield, sugar chains dense enough to help hide parts of the virus from antibodies trying to neutralize it. Mapping that shield precisely, which patches of the spike protein were bare versus sugar-covered, told vaccine designers exactly which exposed regions to target with an immune response, directly shaping how effective a vaccine candidate would be against the real virus. Zoom out to the population level, and the payoff of better glycan diagnostics becomes clear. Liver cancer alone is a leading cause of cancer death worldwide, and much of that burden falls on people whose disease is caught late. Exactly the window a cheap, reliable glycan blood test is designed to shrink. Picture how a future screening pathway might work. A routine blood draw runs through a glycan panel. A normal profile sends you back to standard yearly checkups. An abnormal sugar signature routes you straight to targeted imaging, all before a single symptom ever appears. Artificial intelligence is now entering the picture too, trained to recognize subtle glycan patterns across thousands of patient samples that would take a human analyst months to spot by eye. Early results suggest these pattern recognition models can flag combinations of sugar changes tied to disease with speed and consistency traditional lab review can't match. Put together, mass spectrometry, lectin arrays, viral shield mapping, and AI pattern recognition are turning glycomics from a handful of individual blood tests into a full diagnostic discipline of its own, one built to catch what genomics alone was never designed to see. Let's bring it all together. Across this video, we've traced the glycan code from a single sugar ring all the way to cancer diagnosis, autoimmune disease, and vaccine design. So here's the sweet truth in 5 points. As exciting as this frontier is, it's worth being balanced about it. Glycan testing adds real, complementary information, but it's not a replacement for genomics. And glycan structures are genuinely harder and more expensive to measure precisely than a DNA sequence. Here's a simple way to lock in the core idea. Remember the word SUGAR, structure branches instead of running in a line. Undetected by DNA tests alone, glycans change hour to hour, antibodies and cells wear them as labels, and risk of disease often shows up here first. Quick check before we wrap up. Which blood test we discussed today is based on measuring a fucose-tagged sugar version of a liver protein? Think it through, then let's reveal the answer. If you remember nothing else from today, remember this short list. It covers everything a curious, health-minded viewer really needs to carry forward about where this field stands right now. One honest caution before you go: don't self-diagnose from a commercial glycan or biological age kit without a clinician's guidance. These are genuinely promising tools, but interpreting them correctly still takes clinical context that a home test alone can't provide. So the next time someone tells you your genes are your whole story, remember, there's a second code, written in sugar, changing every single hour, and medicine is only just learning to read it. That's the glycan code, not a replacement for genomics, but the next layer of it, dynamic, information-dense, and already reshaping how disease gets caught early. Here are the key references and sources for this video. Thanks so much for watching. If this helped, like the video and subscribe for more clear, simple explainers, and I'll see you in the next one.